The overall goal of this procedure is to profile gene expression levels of individual human embryonic stem cells using single-cell quantitative reverse transcription polymerase chain reaction, or RT-PCR. This is accomplished by first using fluorescent activated cell sorting to sort individual cells into each well of a 96-well plate containing cell lysis buffer. The second step is to reverse transcribe RNA to generate cDNA from single-cell lysates. Next, the cDNA is amplified by polymerase chain reaction. The final step is to perform quantitative RT-PCR on the amplified cDNA. Ultimately, this assay is useful for studying gene expression in heterogeneous populations such as human embryonic stem cells.
The main advantage of this technique over existing single-cell quantitative RT-PCR methods is that this method is easier and more robust.
Prior to FACS purification, the OCT4 EGFP human embryonic stem cells, or HESCs, are detached from the 60mm dish. Remove all medium, add 1 milliliter of AccuTase, and incubate at 37 degrees Celsius for 20 minutes. Neutralize AccuTase with 1 milliliter of human ES media. Centrifuge to pellet the cells.